Hello friends and welcome. In today's lecture we shall talk about detailed overview of lipoproteins, types of lipoproteins, chylomicrons, VLDL, LDL and HDL and their functions. The main objectives of today's deliberation are to have a general understanding of lipoproteins, to give a detailed classification of lipoproteins and to describe the biological importance of chylomicrons, LDL, VLDL and HDL. To begin with, let me give you a brief introduction of lipoproteins. Fats absorbed from the diet and lipids synthesized by the liver and adipose tissue must be transported between the various tissues and organs for utilization and storage. Since lipids are insoluble in water, the problem of how to transport them in aqueous blood plasma is solved by associating nonpolar lipids with amphiphasic lipids and proteins to make water-miscible lipoproteins. Lipoproteins mediate this process by transporting lipids from the intestines as chylomicrons and from the liver as very low-density lipoproteins to most tissues for oxidation and to adipose tissue for storage. Lipid is mobilized from adipose tissue as free fatty acids bound to serum albumin. Lipids are transported in the plasma as lipoproteins and four major lipid classes are present in lipoproteins. Plasma lipids consist of triglycerols 16%, phospholipids 30%, cholesterol 14% and cholesterol esters 36% and a much smaller fraction of unesterified long-chain fatty acids 4%. This latter fraction, the free fatty acids, is metabolically the most active of the plasma lipids. Now let's try to understand the classification of lipoproteins. Four major groups of plasma lipoproteins are present. Because fat is less dense than water, the density of a lipoprotein decreases as the proportion of lipid to protein increases. Four major groups of lipoproteins have been identified that are important physiologically and in clinical diagnosis. These are chylomicrons. They are derived from intestinal absorption of triacylglycerols and other lipids. Very low density lipoproteins, VLDL, derived from the liver for the export of triacylglycerols. Low density lipoproteins, representing a final stage in the catabolism of VLDL, and high density lipoproteins HDL involved in cholesterol transport and also in VLDL and chylomicron metabolism. Triacylglycerol is the predominant lipid in chylomicrons and VLDL, whereas cholesterol and phospholipids are the predominant lipids in LDL and HDL respectively. Lipoproteins consist of a non-polar core and a single surface layer of amphiphasic lipids. The non-polar lipid core consists of mainly triacylglycerols and cholesterol esters and is surrounded by a single surface layer of amphiphasic phospholipids and cholesterol molecules. These are oriented so that their polar groups face outwards to the aqueous medium as in the cell membrane. The protein moiety of a lipoprotein is known as an apolipoprotein or apoprotein, constituting nearly 70% of some HDL and as little as 1% of chylomicrons. Some apolipoproteins are integral and cannot be removed, whereas others are free to transfer to other lipoproteins. The distribution of apolipoproteins characterizes the lipoprotein, One or more apolipoproteins are present in each lipoprotein. The major apolipoprotein of HDL are designated A. The main apolipoprotein of LDL is apolipoprotein B, B100, which is found also in VLDL. Chylomicrons contain a truncated form of ApoB, B48, that is synthesized in the intestine. While B100 is synthesized in the liver, ApoB100 is one of the longest single polypeptide chains known. ApoB48, 48% of B100 is formed from the same mRNA as ApoB100. ApoC1, C2 and C3 are smaller polypeptides freely transferable between several different lipoproteins. ApoE is found in VLDL, HDL, chylomicrons and chylomicron remnants. Apolipoproteins carry out several roles. They can form part of the structure of the lipoprotein, example ApoB. They are enzyme cofactors or enzyme inhibitors. They act as ligands for interaction with lipoprotein receptors in tissues. Let's understand the lipoproteins one by one. First, we'll talk about chylomicrons. Chylomicrons are assembled in the intestinal mucosa as a means to transport dietary cholesterol and triglycerides to the rest of the body. Chylomicrons are therefore the molecules formed to mobilize dietary lipids. The predominant lipids of chylomicrons are triglycerides. The apolipoproteins that predominate before the chylomicrons enter the circulation include ApoB48 and ApoA1, ApoA2 and ApoA4. ApoB48 combines only with chylomicrons. Chylomicrons leave the intestine via the lymphatic system and enter the circulation at the left subclavian vein. In the bloodstream, chylomicrons acquire ApoC2 and ApoE from plasma HDLs. In the capillaries of adipose tissues and muscles, the fatty acids of chylomicrons are removed from the triglycerides by the action of lipoprotein lipase, which is found on the surface of the endothelial cells of the capillaries. The ApoC2 in the chylomicrons activates LPL in the presence of phospholipids. The free fatty acids are then absorbed by the tissues and the glycerol, backbone of the triglycerides, is returned via the blood to the liver and kidneys. During the removal of fatty acids, a substantial portion of phospholipid ApoA and ApoC is transferred to HDLs. The loss of ApoC2 prevents LPL from further degrading the chylomicron remnants. Moving on, let's talk about VLDLs, IDLs and LDLs in this section. The dietary intake of both fat and carbohydrate in excess of the needs of the body leads to their conversion into triglycerides in the liver. These triglycerides are packaged into VLDLs and released into the circulation for delivery to the various tissues for storage or production of energy through oxidation. VLDLs are therefore the molecules formed to transport endogenously derived triglycerides to extrahepatic tissues. In addition to triglycerides, VLDLs contain some cholesterol and cholesterol esters, and the apoproteins ApoB100, ApoC1, ApoC2, ApoC3, and ApoE. Like nascent chylomicrons, newly released VLDLs acquire ApoCs and ApoE from circulating HDLs. The fatty acid portion of VLDLs is released to adipose tissue and muscle in the same way as for chylomicrons through the action of lipoprotein lipase. The action of lipoprotein lipase coupled to a loss of certain apoproteins, the ApoCs, converts VLDLs to intermediate density lipoproteins, also termed VLDL remnants. IDLs contain multiple copies of ApoE and a single copy of ApoB100. The presence of multiple copies of ApoE enable these lipoprotein particles to have very high affinity for the LDL receptors on cells such as hepatocytes and adrenal cortex cells. Conversion of VLDL to IDL is also associated with loss of ApoCs by transfer back to HDLs. Further loss of triglycerides as well as transfer of ApoE back to HDL converts IDLs to LDLs. The presence of the ApoB100 protein allows LDL to be recognized by the LDL receptors, but the lack of ApoE makes the affinity much lower than that of IDL. The liver takes up IDLs after they have interacted with the LDL receptor to form a complex which is endocytosed by the cell. For LDL receptors in the liver to recognize IDLs require the presence of ApoB100 and is enhanced in the presence of ApoE. The LDL receptor is also sometimes referred to as the ApoB100 ApoE receptor. Although LDL particles possess ApoB100 which is required for LDL receptor binding, the loss of ApoE makes their affinity for the receptor reduced. The exclusive apolipoprotein of LDLs is ApoB100. LDLs are taken up by the cells via LDL receptor mediated endocytosis. The uptake of LDLs occur predominantly in the liver, adrenals, and adipose tissue. As with IDLs, the interaction of LDLs with LDL receptors require the presence of ApoB100. The endocytosed membrane vesicles fuse with lysosomes in which the apoproteins are degraded and the cholesterol esters are hydrolyzed to yield free cholesterol. The cholesterol is then incorporated into the plasma membrane as necessary. Insulin and triiodothyronine T3 increase the binding of LDLs to liver cells, whereas glucocorticoids, example dexamethasone, have the opposite effect. It's now time to talk about the last lipoprotein, that's high density lipoprotein or HDLs. HDL is synthesized and secreted from both liver and intestine. However, ApoC and ApoE are synthesized in the liver and transferred from liver HDL to intestinal HDL when the latter enters the plasma. A major function of HDL is to act as a repository for the ApoC and ApoE required in the metabolism of chylomicrons and VLDL. Nascent HDL consists of discoid phospholipid bilayers containing ApoA and free cholesterol. These lipoproteins are similar to the particles found in the plasma of patients with a deficiency of the plasma enzyme lecithin cholesterol acyltransferase LCAT and in the plasma of patients with obstructive jaundice. LCAT and the LCAT activator ApoA1 binds to the discoidal particles and the surface phospholipid, and free cholesterol are converted into cholesterol esters and lysolecithin. The non-polar cholesterol esters move into the hydrophobic interior of the bilayer, whereas lysolecithin is transferred to plasma albumin. Thus, a non-polar core is generated, forming a spherical pseudomycellar HDL covered by a surface film of polar lipids and apolipoproteins. The class B scavenger receptor B1, SRB1, has been identified as an HDL receptor with a dual role in HDL metabolism. In the liver and in steroidogenic tissues, it binds HDL via ApoA1 and cholesterol esters is selectively delivered to the cells, although the particle itself, including ApoA1, is not taken up. In the tissues, on the other hand, SRB1 mediates the acceptance of cholesterol effluxed from the cells by HDL, which then transports it to the liver for excretion via the bile in the process known as reverse cholesterol transport. HDL3 generated from discoidal HDL by the action of LCAT accepts cholesterol from the tissues via the SRB1, and the cholesterol is then esterified by LCAT, increasing the size of the particles to form the less dense HDL2. HDL3 is then reformed either after selective delivery of cholesterol esters to the liver via the SRB1 or by hydrolysis of HDL2 phospholipids and triacylglycerols by hepatic lipase and endothelial lipase. This interchange of HDL2 and HDL3 is called the HDL cycle. A second important mechanism for reverse cholesterol transport involves the ATP binding cassette transporter A1, ABCA1 and G1, ABCG1. These transporters are members of a family of transporter proteins that couple the hydrolysis of ATP to the binding of a substrate, enabling it to be transported across the membrane. ABCG1 mediates the transport of cholesterol from cells to HDL, while ABCA1 preferentially promotes efflux to poorly lipidated particles such as pre-HDL or ApoA1, which are then converted to HDL3 via discoidal HDL. Pre-HDL is the most potent form of HDL, inducing cholesterol efflux from the tissues. HDL concentrations vary reciprocally with plasma triacylglycerol concentrations and directly with the activity of lipoprotein lipase. This may be due to surplus surface constituents, example phospholipid and ApoA1, being released during hydrolysis of chylomicrons and VLDL, and contributing towards the formation of pre-HDL and discoidal HDL. HDL2 concentrations are inversely related to the incidence of atherosclerosis, possibly because they reflect the efficiency of reverse cholesterol transport. HDL-C, that is HDL1, is found in the blood of diet-induced hypercholesterolemic animals. It's rich in cholesterol and its sole apolipoprotein is ApoE. It appears that all plasma lipoproteins are interrelated components of one or more metabolic cycles that together are responsible for the complex process of plasma lipid transport. With that, we come to the end of today's program. Hope you have understood it well. Thanks and goodbye.